Okay, well, it's my great pleasure to introduce Nancy Morin. Nancy Morin is a council member with the California Botanical Society. That's how I got to know her. She's been a council member for over a decade, so she holds a lot of our institutional memory, and it's been a real joy to get to know her a little bit through the council and get to work with her. Um, she grew up in California and got her bachelor's degree and PhD in botany from Berkeley in 1980 under Bob Ornduff. Her PhD thesis was on Githopsis. I'm probably saying that wrong. In the Campanulaceae, she stuck with that Campanulaceae theme for much of her research career. She had a postdoc at the National Museum of Natural History with Stanwin Shetler, working on seed morphology of North American Campanuloideae. And then from there, she went to the Missouri Botanical Garden, where she was the administrative curator of the herbarium and the editor of the Annals Missouri Botanic Garden. Eventually, she became the assistant director. And in the early 1980s, she was a part of a group of botanists who met to discuss how to develop flora of North America. Um, uh, yeah, north of Mexico. So Peter Raven, who was the director of the garden, invited her to develop the plan for the project. They secured funding in 1986 and published the first two volumes in 1993. This is a massive project, and it's nearly complete at this point. She left the Missouri Botanic Garden in 96 to become the director of the American Association of Botanical Gardens and Arboreta. And then in 2000, she moved to be the director of the Arboretum at Flagstaff. And then she retired in 2004 and moved full-time to Point Arena to continue working on the flora of North America and her Campanulaceae studies. She still has courtesy appointments at Northern Arizona University, at the UC Jepson Herbarium, and the California Academy of Sciences. And today she'll be talking about all the interesting and still open questions in Nemacladus or Nemacladus, depending on how you pronounce it. So thanks very much for speaking tonight, Nancy.
Thank you. So now that you're the host, you need to let me share my screen.
Oh, okay.
I lost the ability to do
That's funny.
that. I know.
Zoom. Okay. Um,
And I really want to thank everybody else for coming. It's just you're here for Nemacladus. I'm going to stick with Nemacladus. But it's just such a wonderful group of plants, and it's so diverse, so interesting, and so small. Um, can I share yet?
I think so. I think I changed the setting, but let me know
Yay,
if
there you
it's
go.
not working.
No, I did. Well, can you see my screen?
No.
Hmm.
Um, are you able to hit the share button? There you go.
Yay. Okay, so I'm going to start the slideshow from the current slide. Yes.
Great.
I don't know how to
That's good.
get rid of this sidebar thing. Wait. We're not there yet. So, I really don't know how to get rid of that. All right. Maybe it'll just go away. Um, this is a wonderful group, and I want to first point out that a lot of the work has been done in collaboration with Tina Ayers and with Lois Neff, and they might be on the call by now. Um, both at Northern Arizona University, and I'm very grateful that NIU has allowed me to have a courtesy appointment still too. So thanks to them. Oh, maybe that's an X. Wait.
Do you need some help, Nancy? What's going on?
I really don't. I'm sharing, but it's not sharing what I wanted to share.
We see the overview slide.
Really?
Yeah.
Okay. So I don't know how to get rid of this. There we go. All right. So sorry, folks. And I can't waste time because I've got way too many slides. So first of all, I just wanted to give you a kind of overview of where it fits in the overall you know, world of plants. Just a few slides on this. It's a genus of about 26 species, and it's endemic to Western North America. Campanulaceae is a pretty large family, and there are 5 subfamilies, and you can see that there are 2 big ones and 3 little ones. Nemacladoideae is the one that Nemacladus is in, obviously. But the other 2 are really interesting too, and very locally distributed and interesting. Campanuloideae has about 1,200 species. The flowers are radially symmetrical. They are not resupinate. The corolla lobes are usually fused part of the way. Filaments and anthers are free. Pollen is held on hairs along the outside of the styles. So here's the style, and that's all bunches of pollen hanging onto it. And then that falls off, or it gets taken away by pollinators, and the stigmatic lobes open up. And the filaments and the anthers are free, and they just kind of fall away because they don't need to be there anymore. And they have an inferior ovary. Lobeolioideae has bilaterally symmetric flowers that are generally resupinate. The corolla lobes are usually fused part of the way. The filaments and anthers are fused into a tube. The pollen is presented on the tip of the stigma, which pushes its way through that tube. And the ovary is inferior here too. I love this. I'm not sure I will have managed to acknowledge all of the photographers. This might be a Jerry Carr photograph. It's such a good photograph. Anyway, shows that you can see that really well fused filament tube and anthers, and the stigma starting to push through. Can you all see my cursor as I'm moving it around? Is anyone unmuted who can tell me if you can see the cursor?
Yes, we can see your
Yes,
cursor.
yes.
I
Oh,
can
good.
see it.
Terrific. Thank you so much. Okay, so on to the good stuff. It's really weird within this family. The flowers are both, some of them are resupinate, some of them are non-resupinate. And resupinate has, just to help you, because I'm spatially dyslexic and I always have to remember this, but whoops. 2 petals up is resupinate, like you see in most lobelias. 2 petals down is non-resupinate, and the flowers twist on the pedicel. They can have nearly radially symmetric flowers, or bilaterally symmetric. The lobes are nearly free, or very well fused. And some of the species have superior ovaries, so that's pretty different. This is from Crowell, oops, 2016, just to show you where they thought that the Nemacladus came off, and it separated out about 60 million years ago. So here are the 1,200 or so lobelioids and the 1,000 or so campanuloids in just this little tiny branch. So let's talk about those flowers. Here's Nemacladus rubescens, which is probably the showiest one, the one that catches people's attention the most. And you can see that it has the filaments partially fused into a tube. They start to be a little separated at the base, but they also have this pad of tissue and these single-celled hairs that are filled with liquid attached at the base, and then really quite enormous nectar glands at the base of that. Tina and I, when I first started working on it, part of my quest was to get photographs of everything because you can't tell any of this from the herbarium specimens. And then she and I started doing SEM of the, whoops, SEM of the flowers. And I think everyone should do this because it's so amazing what you can see and the detail that you can get, and especially on these. So this is the same, looking at the flower the same way, the cells, the attachment, the filaments. And look closer, and there are these papillae at the base of the filaments. There's papillae all over the nectar glands. And you can see that these cells just kind of collapse in the process. This is maybe a stacking photograph that Lois Neff took, showing again in kind of real life, this is what these look like. We still don't actually know what they do or what the point is, and we can talk later about some conjecture about what that is. But now backing back a little ways to talk more about the general way that Nemacladus works, has a little basal rosette, and there's diversity of what the leaves can look like. We're still looking at Nemacladus rubescens. Here's what the little capsule looks like with its partially, mostly inferior capsule. And you can see that these plants are really branched. Rubescens stays green for a lot longer than most of the other species. You come across these leaves early in the season, and they stay green right through flowering. So that's kind of different than most other species. When I'm showing you maps, these are either coming from the Consortium of California Herbaria or iNaturalist, and they're really just meant to give you a general idea of where the plants are. I've curated the iNaturalist images pretty well, but I have not managed to get in to annotate many of the specimens over the last, whatever it is, 7 or 8 years. So there may be some things that are not correct, but that's where they're coming from. So a group of plants that used to also be considered rubescens is Nemacladus tenuis, and that is split into 2 varieties. tenuous variety. Tenuous has these little kind of triangular upright upper petals, and variety alliformis has these really amazing wing-like upper petals. And the tricky business is that there are intermediates. You can go anywhere from this to these are longer and curvier. So it's not an easy separation or determination to make. Um, and here's what they look like close up. Um, the big cells, kind of donut shaped uh, nectaries. You can see it's apparently pretty unusual that the anthers flip back like this off the tube. Um, and this is work that Jenny Richards did, plant morphologist, and it shows that these things are actually on very long stalks that I guess kind of bend around so that they can all get packed in. And you can see that the nectaries are really sitting up on little um, structures. So I have been trying to figure out if there are any other ways that can define what variety tenuis is versus variety alliformis. And looking at all of the iNaturalist photos, almost all of them, all but like one or two, had no yellow on their petals. So I was thinking, well, maybe that's a tell. Maybe that's how you can distinguish them. But then I realized that even in the paper where I made this combination, I had these images from Aaron Schustaff with bright, bright yellow tips on tenuis. So that's not much help, but his beautiful photographs show how these cells can splay out and be very showy. And then, still thinking about it and looking at those images, I came across these with these funny little patches on the petals. And I asked Jenny Richards what she thought, and she said, well, that could be another kind of nectary. So multiple ways to attract pollinators, if that's true, I guess. And I also came across this little guy. And I think Fred and Carla are on the Zoom with these little tiny short upper petals. So how small can those get ultimately? Um, and and this whole talk is just a shameless ploy to get you all to go out and look and photograph and enjoy. Um, so this is what tenuous basal um, leaves look like. And I love it when people photograph capsules. You can see the capsule opening up here. And here's generally where tenuis variety tenuis is, and this is where those little short upper petals, where that plant was. And this is where variety alliformis is. The plant that you're actually most likely to run across if you're out looking for plants in the desert is Nemacladus orientalis. And so I'm not going to talk much about it, but this is what it looks like, and this is what it looks like close up. It doesn't have all that papillate material here, but it's got all the parts. But often collected as part of the same specimen is this little plant, which I it it has been identified as Nemacladus sigmoideus, but it's often on sheets with orientalis, and when we had good photographs of it, um, I concluded that it should be described as a species with Tina, and it has these wonderful flanking sepals that frame the flower. And I think sepals probably don't get enough credit for how much they add to some flower displays. You can see it's got the little cells here, it's got the filaments. So I really love this plant. This is Nemacladus morfieldii. It has typically quite thin pedicels that kind of droop and are curvy. Sometimes they're less curvy. And it could be that there are intermediates, but between it and orientalis. Here's where orientalis, here's where orientalis is. We started out thinking that morfieldii was pretty limited in its just distribution, but now as people have been looking for it more, it's looking like maybe there is more of it. And this seems like a good place to mention Nemacladus rigidus, because it's another one with not resupinate flowers, the maroon markings, but it's got these wonderful chunky plants. These are the capsules, and sometimes it's like the whole thing is nothing but big chunky capsules. And it's got beautiful flowers when you get close to them, and it also has got these large flanking sepals, and you can see the large cells, the glands, the stigma with the pollen on it, and maybe these little hairy areas could be other nectaries. So switching gears a little bit, now I want to look at the longiflorus complex, which is longiflorus breviflorus, which used to be in longiflorus, and a completely new species, richardsiae. This can be pretty showy, actually, in the context of Nemacladus. This is longiflorus. It's got this wonderful long corolla tube. There's the anthers flung back on the stigma. It actually has blue pollen and these great splotches on the backs of the petals. Here's what it looks like with SEM, and you can see that it just has these little tiny cells on this little flap of tissue up at the top of the ovary. Longiflorus has superior ovaries, so that's unusual. It does have a little nectary at the base of the ovary, and Jenny tested and found that it does produce nectar. Wayne Armstrong did this wonderful photograph diagram to show you the parts. Again, the superior ovary, the gland, and so on. Breviflorus can be really tiny. I mean, barely bigger than any of the pebbles that it's growing in. Has these wonderful basal rosettes that are very fuzzy, can be really curly with the axis very zigzagged, and the pedicels curvy. It's got a short tube, and otherwise it's got all the same parts. So here's the next thing to be looking for, because go away. There's this odd disjunct in the coral pink sand dune and white cliff area in Utah. Here's where here's where Breviflorus mostly is. There's another disjunct in the southern Sierra Nevada near Lake Isabella. I think these two specimens may be some kind of historic artifact, maybe not, maybe a misidentified or mislabeled specimen. This is what the pink coral pink sand dune plant looked like. This is Walter Frutag's photograph. And it kind of has the look of, I mean, maybe it's Longiflorus, but anyway, it would be fun to find it again and to get more of it and see what's going on. And then the big surprise for me was when Tom Chester told me about a locality near Lake Lee. And when I went there the first time, there were these plants, which had a corolla shape that's unlike any of the rest of them, really. And here's Longiflorus just to compare with this flaring open corolla. So I'm hoping people will be on the lookout for that as well. Most of these are historic. I think that's Lake Lee. But recently there have been people finding it near a power plant there near did I write it down? Yes, East Highlands, north of the Santa Ana River. So, and quite a, it's a pretty big population of them. So I think this probably needs to get to be listed as rare, and have some folks finding populations and counting individuals so that we can see how much of it remains. Then another discovery was 2010. Steve Matson sent me a photograph, and I thought, that's different. Jim Moorfield sent photographs. He said, let me know if you find any more. So he managed, I think, to send me a little specimen, but it wasn't any more than this. It was about like that. And then in 2019, Zoom, there it is, big populations of it. And I don't know that anyone's been finding it since then. And this is what it looks like. The most distinctive thing is that it's got these pink stripes on the petals, but these capsules are also pretty distinctive and sort of chunky the way that Rigidus is, flat bottomed, which is unusual. So I'm sure people are going out and continuing to look for it to see if it will show up again. But there were, I think, thousands and thousands of them, and then pretty much gone. They grow in these really rocky areas, big rocks, and I think this is actually an elongated stem below the basal rosette, and then you start getting roots at the bottom. So this happens in a couple of other species as well. Really is adapted to growing in these rocky outcrops. or scree. Again, we thought it was probably pretty localized, but it was found in there were quite a few sightings of it. This was a collection made in the early 1980s by Nancy Fiverette and Holly Forbes while they were doing a fish slough inventory. And another fun find was Nemacladus calcaratus. So I looked at this collection made by um, Barbara Erder. She was with Jim Shivak, who had collected in this area in 1985, and he picked up some of these plants. And then they did a big group collection in 1986. And then there were not any. And then there were plants that were seen. I found them. I went back and looked and found them in 2008. And Duncan Bell collected it since then in 2015. So it's not common. It's not very wide spread. And it's got the distinction of having this little nectar spur, which has got the sepal cocked back. And here you can see the nectar spur and the sepal. And this is what the cells look like, pointy little guys on a pad of tissue. And again, you can see it's got its little long stem down here going into the rocks. And here's its general location. So most of these on the edges here are very recent. So I'm glad people are looking for it. All right, so shifting to another group of plants, we've got Nemacladus interior, montanus, and capillaris. And boy, they really, interior and montanus look very, very similar. They're hard to tell apart if you didn't know where they came from or when they were collected. But interior tends to bloom late and stays green. Montanus blooms early. It has kind of maroon brown stems. And there's no mistaking capillaris when you see it. So here's what montanus looks like. Got the little short cells here and lots of papillae and glands. And interior, pretty similar. And capillaris, which is, maybe, I don't know, 3 or 4 millimeters in size, but it still gets to have all the same parts. So it gets to have a couple of little cells and 2 biggish nectaries and maybe a little lump there. So capillaris is in the Coast Ranges and the Sierra Nevada foothills. Mostly it doesn't get too high usually. And interior is in the Sierra Nevada, and montanus is in the Coast Ranges. And then there are these 2 places where it could be intermediate plants. It's not clear what's here. This is Ortigalita Peak, and lost my cursor. This is, I think, around Hancock. So I would love it if people would look for those. And it's not too far from where the banquet is going to be, so you could, make a trip of it. And then we have, how am I doing on time? Well, still lots of time. these two notable disjuncts, Californicus and Twisselmanii. Californicus is, I'm not sure what's going on up here. That might be an O'Donnell. I don't know if he's on the Zoom. Um, there's this group of populations and then this group of populations. And it would be interesting to know if there's any bridging there. They aren't exactly the same morphologically. Um, here's what they look like generally. So Californicus is interesting because the capsules are circumscissile. They just have a little lid that pops off. And the flowers are fragrant, so that's different. Um, and they have these little hand-like pads with little stubby cells on them and a huge head of on the stigma anther. The other thing that's different is that they form a little rosette and then put out a little stem that hugs the ground, and then they do another little rosette. So that's unlike any of the other species. This is from Cushionberry Canyon. This might be a Aaron Schustaff photograph. I kind of ran out of steam at the end with what to with putting photographers' names on. I apologize. And then Dieter Wilken found little plants in Apache Canyon in Ventura County. And they're just darling. They're huge flowers, little teeny tiny plants. I love this photograph because it shows this beautiful stigma and the anthers, and that there are hairs at the base, at the tip of the style, or the base, the tip of the filaments, I guess it is. So I'm not sure what's going on here, whether these should be recognized. I haven't really been able to settle on other morphological characters that might make sense to describe them as separate taxa. And a similar sort of thing happens with Nemacladus twisselmanii. So this was the historic locality of twisselmanii on the domes in the Pine Flat area and beyond. Only seen every, I don't know, 10 or 15 years, whatever anyone see them. They're very, very tiny. They do the same thing as Californica, which is they throw out these little stems and then have another little rosette sort of thing. That looks like it's just a stem that just fell over, but they do actually do the rosette thing. Here's what the flowers look like. So in these two different populations, this is Lois Neff's the genetic tree. Sorry, just don't know how to get rid of this other stuff, but I can find it. Um, there's the West Ventura County locality, the Apache Canyon, and for Californicus. Here's Twisselmanii, and there's another Twisselmanii somewhere up here. So that's pretty distant. They're that, hundreds of miles away from each other, and it would be good to know what to do when they're morphologically so similar. Um, how to think about that. So if you're out and about, the Southern Sierra Nevada, I think definitely has got a lot of things still to be discovered. It would be fun to do more field work there. The folks at Rancho at California Botanic Garden have been doing a great job of doing uh, local floras there. The Pinochet Hills is another area that I think is going to be hiding some other new taxa, undescribed taxa. The Tehachapi Mountains, I think, is another place to be looking for plants. I kind of have dreams about how to go about figuring out what is going on with all these structures in the flowers, and documenting what pollinates them, how it all works, whether they have a do those cells have some kind of mechanical purpose? Do they reflect light? Do they moderate the heat? Um, Usually you don't see them tune off, so I don't think they're food things, but it would be so much fun to figure that out. And there's definitely a diversity of insects that visit them. It looks to me most of them just kind of hang around. They just are there hanging out. So figuring out how what they're doing, what pollinates it. I wonder if some of them might, like the longiflorus, perhaps could be pollinated at night. Um, Still lots of things to figure out there. And if you're going out, here are a few tips. You have to really look down. And if you're already down, you should look sideways. Many of us see them by seeing their shadows or the flowers bouncing in the breeze. Um, Several species can grow together. If you found one, you should keep looking for a while, and maybe there will be more. And I think that's it. Actually, I thought I would be way out of time, and I guess I talked faster than I expected. So thanks very much.
Thank you,
Happy
Nancy.
to answer questions. And I didn't talk. I mean, there are more species. I did not talk about the species that are in the Sigmoideus group, which ended up being 4 or 5 other species. So um, there's more there. I didn't divulge everything. There's still more.
Nancy, can you see who has their hands raised? Or do you want me
Yeah,
to call them?
Jim
Okay.
Moorfield.
Hi, Nancy.
Hi.
You had mentioned earlier about not being sure how Nemacladus inuensis was doing. Um, I can just update you and the whole group that it's doing great. It seems to come out just about every year, probably helped by the fact that it's a higher elevation species up in the pinyon juniper, so its precipitation is a little more reliable than down in the deserts. And it's shown up as far north as in the Mono County in the White Mountains, all the way down to the southern Inos, the Panamint Mountains, and in Nevada in the Silver Peak Range, thanks to Perry Lee Pipkin's floristic work out there. So it seems to be doing fine.
Do you think it's was there all along and people just didn't notice it?
I think so.
Because there really are not herbarium. There are not collections. It's not like it was hiding in the herbarium and nobody recognized it. I don't think it was there.
Well, your work and the advent of digital macro photography, I think, has changed our perception of a lot of the distribution and abundance of these plants.
That's great. Thank you. And thank you so much for your work. Really.
Oh, sure, it's all good fun.
It's good fun indeed. Peter?
Um, yes, what a interesting and challenging little teeny plant group. I mean, how do you have a sense of how well at this point the species and their distributions are known since um, there's they're in these inaccessible places and they're so small.
Well, 8 of the ones that I think that I talked about were I just recently described. So it's you have to really get in there and look at them. And, earlier botanists didn't have a way to do it. And they were just working from herbarium specimens, and you couldn't tell a lot of the stuff from the herbarium specimens. So, but being small, does it does take more work to figure out what's going on. Wayne?
Thank you. A great talk. Really appreciate it. I was part of the fish sleuth study where the Nemacladus was found. We worked out there for a number of years, mostly in the alkali where many rare plants occur. But this was on tableland, which seems a very different habitat than uh, pinyon juniper. So I just wonder, as I one collection out there at Fish Slough, maybe something to think about. And I had mentioned when we were talking before that the first Nemacladus ever found was Ramosissimus in northern Santa Barbara County. It's a great plant, but you didn't talk about it because it's in a different group. So,
Right.
but I happen to really like that plant. So anyway, thank you so much.
Thank you. Thank you. Andy? Do you want to unmute yourself or whatever?
Hi, yeah, so great talk. So I just going to reread my comment question. So I'm wondering how old the Nemocladus and the radiation that you were looking at is. To put this into context, I looked up the fossil record of the Campanulaceae, which are being a largely herbaceous family. They don't have much of a fossil record, but they do have pollen. And the oldest pollen is from the Oligocene of Poland, some 30 something million years ago. So, which is a lot younger than the late Cretaceous date that you threw up at the beginning. Can you can you say anything about how long you think the Nemocladus radiation in the Western North America has been, and what might be driving it, and what geological or climatic events might be responsible?
I don't know enough to hazard even a guess, but it seems to me that Tina and Lois are probably on the call, might be on the call. Do you want to say anything? Um,
thought I thought I had something in my thesis about it, but I'm not finding how old
okay.
it is.
I talk at the botany meetings in in this last year. I think it was this last year. Suggested that the lobelioids came in through the or lobelia came in through the mountains in the Sierra Madre Occidental in Mexico. And Pseudonemacladus, which is the only other genus in the subfamily, is a perennial shrubby sort of plant. Not really shrubby, but anyway, perennial. And it has very unusual flowers. They're Nemacladus-like, but anyway, it's complicated. And so it could be that there was a radiation from there, and the precursor to both groups, Nemacladus and Lobelias, split off and made their way up. It looks like the most widely distributed is Nemacladus rigidus through the Great Basin. And I don't know, a lot of time passed. Things changed. They had nobody else to breed with. They had nobody but just the genes that they started out with. So um, I don't know. Andy, do you want to hazard a guess?
I don't know. I'm just thinking. It looks to me like it's a very from. I have of course seen this plant, or I don't know which species, but all the photographs that you've been throwing up have been very familiar. It's an extremely dry, adapted, xerophilic thing, and
Mhm.
it makes me imagine that. It makes me imagine it's probably quite recent, with they're probably not much older than the formation of the California floristic province, or at least the type of coastal mountain sort of flora that characterizes it. So I'm guessing it's pretty young. Now,
But
the lineage may be ancient, but the radiation, I'm guessing, is 10, 15 million years at that, if tops, just based on the
okay,
ecology that I'm seeing.
so Wayne, did you have something you wanted to say? And maybe I'll pull up that early slide that shows the crowl at L.
Sorry, I guess I didn't take my hand down, but thank
Oh,
you.
Yeah, I don't know. I don't know. I'm guessing. That's why I was asking you the question. Great talk.
thank you.
Nancy, Lois in the chat says Krell suggests 44 million years ago, but I'm not
Hmm.
sure if that's for the Nemacladus radiation or one of the deeper nodes that you were talking
No, I
about.
think I think that it shows Nemacladus coming off at 60. So
Okay.
then other species within it would be coming off
And then there's
later.
another question in the chat from Laura Yatim. I'll just read it to you. Nemacladus seems to appear and disappear from sites somewhat unpredictably. Do you think this pattern is mostly due to how small and easily overlooked the plants are, or might it reflect a long dormant or persistent seed bank? Good question.
Oh, I'm pretty sure it has a long life seeds. And um so an example would be Twisselmanii in variety Twisselmanii in the southern Sierra Nevada. Really, people, because it's so rare, people in the Fish and Wildlife Service and others go out, you know, every year hunting for it. They know exactly where it ought to be. It's like the size of someone's living room, and they don't find it, and then it comes up. So I think the seeds must be long lived. Um, but obviously, um, a good rainfall year is going to mean that there are more to be found. Glenn, do you want to unmute?
Could you review that early slide showing the divisions in Campanulaceae and how it fits in to that again, please?
Yeah. Let's see. It doesn't want me to go back. I don't know how to do that. No, Glenn, I'm sorry. I don't know how to do it.
It's okay. Thanks for trying.
I had a
Okay,
question.
Ann.
Oh, can I ask a question about the pollination biology?
Mhm.
It sounds like nobody's really done any studies on pollination. Is that right?
That's right. The only thing that I did when I was in Flagstaff had greenhouse plants growing, and I did some hand pollination and some exclusion, and the larger um, like orientalis and rubescens would not self-pollinate.
Okay. The
I
little
mean,
ones would though?
the little ones would very consistently. And in fact, when you collect them, you you put them in the press and you think, oh, good, I've got great flowers. And when you open them up, you've got capsules. So they are taking care of it on their own. Anyone else?
How far north do they go, Nancy?
Rigidus gets into Oregon, into eastern and kind of east and central Oregon.
Well, I guess I need to go further south because they are fascinating creatures.
Yeah, you and take some of your photographs.
Well, I don't see all those tiny things until I put them on the computer.
Yeah, yeah. So, I would really like to thank everybody who's taken the patience that is required to get a really good photograph of Nemacladus flowers. It's just unbelievable. And even though cameras have gotten better, it's still, you got to hunker down. You've got the wind. It's very, very difficult to do. And people have been taking fabulous, fabulous photographs. I'm so grateful, and I want you to know that at least someone looks at them really carefully and with tremendous gratitude.
Well, it's all good fun.
It is.
Well, it's probably the right time of year to get out there and look for them.
Exactly, which is why I wanted to give the talk this month. I want y'all to go out and then see, when you see them, even if you're not able to get really up close, in your mind, you will say, oh yeah, I know what that looks like in there. I know what those flowers look like.
And once you see them, then you see them.
Yeah, once you see them, it's what is the word?
Calibration.
Yeah. Mm-hmm.
Based on the weather
Okay.
forecast, everyone needs to hurry. So they're gonna see them this year. I'm sure
Yeah.
they're out, but they're gonna go fast.
Well, and the interior ones are, I mean, they keep going. I pretty much was not finding some of the species that I didn't talk about until I would find them Mother's Day weekend, because I would go south to visit my mother and go out and look for plants, and that's when they were blooming. And they can go even further than that. The ones in the southern Sierra Nevada are quite high, so there's still lots of good time.